Is it better now?
Yeah, but still not great. I have to see an orthopedic surgeon end of the month.
Oh, shit. Okay.
That doesn't sound good.
No, yeah, but okay.
But I hope I just have coffee with that person and they say fine and I go.
Fine, you can go. Yeah, yeah.
My plan.
Yeah. You see. Okay.
There's a slim chance a surgeon might have a different plan, but
Okay. But you can walk. Yeah, it's
yeah, I can walk again, just
Yeah.
not for too long. That's the only thing.
Okay.
Hey Anna.
Hello.
And that's like the vicious cycle. It works better, so I walk more and I overdo it, and then it hurts even more.
So you stop, and after you need to redo it. Yeah.
Yeah. You would think at my age you learned how this works, but no. Right. That is looking better. You guys had me worried. I was sitting there at 9:30 by myself. I'm like, did I miss something? So thanks for coming.
And yeah, a second guy. Yeah, two bald guys. Yeah.
Boop, boop. We are complete. You're even in the same city now.
Yeah.
Kinda.
Exactly. Yeah, I found it. No, he's not here yet.
Chris, is that the fridge behind you? Do you have a fridge finally?
I do now, but it's not the fridge, fortunately.
Oh. Congrats on the fridge.
What a random opener for a Monday morning.
Well, Chris is settling, so he gets all the deliveries day by day, and there's like every day something exciting because some like new item, new furniture is delivered. So
knowing Chris for a while, I can tell you that's not him settling. That's just him.
It's always like that.
There's always deliveries for him. Right, let's make a start. Thank you, Anna, for hosting last week when I wasn't here. It's always stressful to host and present at the same time, but you did a fantastic job, especially considering that technology let you down. So today, let me get the link first. See, multitasking, not easy. Today, I thought we could discuss a paper that I came across and thought it was absolutely exciting, and then started digging a bit deeper and realized it's even more exciting than I thought it was. And I came across it so I was teaching an anatomy class 2 weeks ago, and they already updated their booklet with a new meningeal layer. So there is now 4 meningeal layers, and they're already teaching that to the new students this year. So they asked me questions about this layer called SLYM. I'm like, what's going on here? I don't know the structure. So after class, obviously, I was like, okay, what is the SLYM thing? And there was a paper this year that came out, I shared the link, that described a new meningeal layer in the brain. It came out in January. Now, there's a couple of things I think are exciting about that. Number one, it's a pure brain anatomy paper that got into Science. Boop, boop. Number two is that anatomy is still changing. So it's not as static as many people think, especially when they start learning anatomy. Number three is we potentially have to update textbooks, which is always exciting. Now, let me show you. Little disclaimer, I don't actually know how to pronounce it, but it's spelled like slime. And when I first put it into Google actually gave me a lot of pictures of slime, the kids' toy. So I just call it slime. But this is a depiction of what it looks like. So you have can you see the mouse? Yeah, okay. So you have going from dorsal to more ventral, you have the skull. Then underneath you have the dura mater. And for those of you who've seen it, it's like a super solid sturdy meninge that you can't easily rupture, like you can really pull it and it holds. Underneath, you have the arachnoid mater. Then you have the subarachnoid space, in which you can find in green the new SLYM. And then you have the pia mater that is really neatly tucked into the brain gyre and sulci. Now, in terms of nomenclature, dura mater means something akin to hard mother. Arachnoid is inspired by cobweb because you have this cobweb-like structure in there. And then pia, I actually don't know. But what you can see is that the dura mater is like a cover over the brain. Looks a bit like a tent. And you don't actually see what the brain looks like underneath, whereas with the arachnoid and the pia, it's kind of see-through. You can see it through, and when you want to peel it away, it's like really soft as well. Now, the purpose of these membranes and meninges is to keep your brain in place. So number one is that layered and adhesion structure. Make sure that you are attached with the skull to keep in place, but also you have the corticospinal fluid that runs through it, and the meninges are important to have that flow and transport of CSF in the brain. Right, this is the classical idea of the meninges on top of the brain, their structure and their function. Now, this new layer, as I'm showing you, called SLYM, actually is short for subarachnoid lymphatic-like membrane. And it turns out to be a highly debated structure in the human brain. So I mentioned that they published it in Science. And the paper is not easy to read if you don't know immunology and staining and all those things. Because it's just a lot of acronyms. But what's very interesting is if you go into the letters, so if I show you briefly, this is the paper. And here you have 9 E letters. And you can already see concerns of rigor and objectivity. It's very long. Sorry, I was kind of got stuck reading it. It's a bit like a, it's easier to read than the paper, let's put it that way. But they raise a couple of good concerns in the comments, so it's worth having a look at those. But it's not a new discovery per se. So the idea of the meninges maybe being more than 3 has been around. So here we have a series of papers, the structure and functional features of central nervous system lymphatic vessels. We have the junctions in the meninges and marginal glia. We have avenues for entry of peripherally administered protein to the central nervous system, mouse, rat, and squirrel monkey. We have the meningeal compartments of the median eminence of the cortex and comparative analysis. And we have a review on the question of the subdural space. So you can see that this idea has been around since the 70s and it keeps coming back. So this is the latest iteration of this debate. Now, what did they actually do? And I tried to do my best to explain it to you. But here is what you can see. So number one is looking at the anatomy of the meninges. So what you see here is the dura mater. Then you have the subarachnoid lymphatic layer. You have the vessels and you have the cortex underneath. So what they did in the first investigation was to see what the structure is, what it is made of. So they used in vivo 2-photon microscopy And this was all done in rodents and humans in the end. And then something called harmonic generation to visualize the collagen fibers and the vasculature. Now, the result of that is that they could describe SLYM as an independent membrane or layer in that space. And then the next question was, well, what does it do? So one thing they were interested in is whether or not this membrane is permeable to proteins, for example. So here's what they did first. They looked at the different layers. They injected proteins that were smaller than 1 micron, sorry, larger than 1 micron, and checked if they actually changed their location and went through the SLYM layer. Now, the result of that is that many solutes in the CSF, so the cerebrospinal fluid, such as the cytokines and growth factors, are considerably smaller than 1 micron in diameter. And the result was, like, they couldn't say if the smaller particles went through the layer, but the larger particles did not. And that's what you see here. So they basically injected particles and just followed if they pierced through the membrane or not. Now, the next question was then to see if smaller particles can get through. And what they basically found was that that membrane is a good separation and it is impermeable. But obviously, if there is a leakage or dural damage, then particles can get through. No surprise there, I would argue. But they actually checked for that. Yeah, so there's a lot of methods and abbreviations, as you can see here, that for people with our background is hard to grasp, but I thought it was utterly exciting that there's a discussion about a new anatomy in the brain that made it into science. And that's all I had prepared. Happy to discuss the paper and your thoughts and ideas. Hands are already up. Great. Chris.
So, so you said that it was kind of discussed if the paper was actually, the paper's claims were actually correct. So if it's correct, I mean, I don't know anything about neurosurgery and stuff, but I would guess that there would be some implications when you open someone's skull, you operate on the brain, and potentially, because if it's a layer that protects the brain from big molecules, that means that it probably protects it from bacteria. I don't know if viruses are, I don't know if they are too small to be repelled by it, but at least bacteria. So yeah, it would have probably an impact on neurosurgery, and then I guess I don't know if other diseases. I mean, that's not the topic, I guess, but I don't know if other diseases could disrupt this layer, and then that could cause some infections or stuff like that. That could be the cause of some infections, potentially.
Yeah, so the very good points that you raised there. So the idea was that it permits the exchange of very small solutes between the CSF and the venous blood, and as such, it might actually offer us some insights into brain immune barriers and fluid transport in the brain, and also about the transportation of CSF, because we have a fairly good idea where CSF is produced and how it travels around the brain, so from the choroid plexus to the ventricles, la-di-da-di-da. But what is less well known is how CSF is transported within the subarachnoid space. So these are the two or three potential implications of this research, that we can potentially understand better how CSF travels in the subarachnoid space, get insight into the brain immune barriers and transportation, and that exchange of small solutes that then can be used, for example, for administering drugs potentially. So yeah, very good point. And I did say it's a controversial paper because if you, hang on, let me just do that for you. Here we go. So if you open Wikipedia, it already has a Wikipedia page, super quick. And it actually starts with, it's a highly debated anatomical structure, which is not the opening line you want to have for your discovery.
Well, at least you have a Wikipedia page. I don't know.
I mean, you can make your own Wikipedia page, but I don't think you would start it like this. And then, as I said, if you go to the actual paper and scroll down to the E letters, there's already 9 of them. And a common thread that I got from kind of reading through it a little bit is that the authors of the original paper seemed quite reluctant to take on criticism. Some of the critique is like it has been described before in another way, but so that would be lack of novelty. Then, as I showed you earlier, there was one of them that questioned the rigor and the methods. Here there are no separate functional compartments, la-di-da-di-da. So it's fiercely debated underneath the paper in the post-publication reviews. The surprising thing to me was that I already saw it in a textbook for the students without any comment of this being new or this being debated and still to be confirmed in independent data. It was literally just a bit like this figure that I showed you. So they had this figure in the textbook. It was like, boom, there's 4 meninges. I'm like, is that I don't know. But that means that if we teach the anatomy without the body of debate around it, then the next generation goes out being convinced there's 4 layers without ever critically evaluating that. Evie.
Yeah, so and a following question is, did they already link it, link this new structure to some diseases, disorders, or some stuff related?
Not that I'm aware of, but I have to admit at some point I stopped reading towards the end because my head was buzzing with acronyms, and I prepared this at like midnight yesterday, so it's like I can't take it anymore.
Okay, because
so it may be there, but it's definitely not mentioned in...
Okay, so because they are not even sure about this function, so it's not clear what that does. No, or is it clear?
It's somewhat clear in a sense that they looked at the anatomical composition of that layer,
yeah,
and the permeability to see what can get through and what cannot get through. But yeah, as I said, you can see there's no highlights here on page 4 anymore. So whatever happened on page 4.
So if you remove it, what is happening?
That's a very good question.
Yeah.
I don't know.
Okay.
I don't know. I do not recommend that study in humans, just in case. Any other questions or comments?
If there are no others, if I can just comment on the whole thing, that's actually really interesting thing with the textbooks, with the debate about it. I think that first that shows that science is moving, but also that shows that scientists are also attentive. Of course, a paper in Science has more visibility than a paper in other potentially smaller journals. But still, like, you see that scientists are attentive to the validity of what's published, but also you see that there are some people who don't super care about the validity and just immediately take it for granted. So that means that we need some changes, but at the same time, that means that science is at least doing its job. The world of science is doing its job of criticizing everything, but not everything in a blind way, but everything in a careful way, I think.
Yeah, I would absolutely agree. So part of me was like, wow, this is extremely fast impact from a research study to being available in a book. Now, that said, it wasn't like a fully edited, printed book with a publisher. It's the reader for the class, but still, like, that's we would all dream about an uptake of our research that fast, to be taught to the next generation as this is the truth. But what did surprise me was that it was not questioned or discussed, and especially with extremely novel findings. That I mean, you have to at least put the caveat out there that sorry, I was reading what I'm speaking. The caveat out there that this is novel research and has to be validated at some point. But at the same time, I mean, we're all aware of classes where we were sitting and there was like, this knowledge is so old. Why am I sitting in this classroom? So
yeah, but as you said, you can say that it's novel.
Yeah.
It doesn't cost much, you know. You can say, well, recently there's been this discovery that might be a new layer. That's it.
Correct. But I was impressed it was already in there. So the question that just threw me is, I guess, is not visible to the naked eye. Have you tried to dissect it in the anatomy course? So even the 3 layers that we have are hard to separate by eye. You definitely see the difference between the dura mater. But then with the pia and the arachnoid, it's very difficult to see the difference, let alone a layer in between those two. So it's not like you open it up and you're like, oh yeah, how did we not see this for 100 years? And as you can see in the paper, they used a lot of staining methods to kind of flush it out. So it's not visible to the naked eye. Have I tried to dissect it? No, not yet. I was looking for it. I was like turning this. I'm like, where is the slime thing? But yeah, it's not easy to see. Cool. But there you go. There's always new things to do and anatomy to discover. It's never boring when you go to the morgue. There's always, you learn something new with every single brain that you look at. So quite exciting. Right. There's no other questions that I can see. Then thank you very much for joining us this morning. I hope you have some time to read the paper and the comments. It's actually exciting to see that debate evaluate, and bear in mind that this is standing on the shoulders of giants, so to speak, with a tradition that goes back at least to the 70s, with a discussion about what's going on in the subarachnoid space in our brains. With that, have an exciting week in science, and next week I think Leah is presenting. Sorry, Leah. Lilith. And I see you Monday. Bye.